C'est bon. Je peux commencer. Ok. Ah. Okay. Professor Casper? It's over.
Hello, Prof.
Okay. We want to start the defense of the thesis of Madame Mary EMERAGHI. Madame Mary EMERAGHI,
Yes.
we will give you 40 to 45 minutes to tell us the main parts of your work, your thesis. You can start.
Thank you. Thank you so much. I'd like to share my screen. Good morning, esteemed jury members. Good morning, my professors, my supervisors, my colleagues, my friends and family, and welcome to the defense of my PhD. I'll be sharing on my thesis title, Current Status of Maize Streak Virus Disease Incidence and Field Response of some Selected Resistant Hybrids in Benin, in the Republic of Benin. I'll go through the different parts of my PhD, which includes 5 chapters, and the key points, including the background of the work and the problem statements, justification of the study, questions, objectives, and hypotheses. I'll move on to the state of research on maize streak virus disease in Africa. That was in a way to review on what is and what is supposed to be going forward for maize streak virus research in Africa. And thereafter, I would share about a countrywide survey done in the major belts of Benin, and to share our discovery with the work. Then also, I would go into an assessment study of the yields, analysis of the G/E stability of improved maize stop cross hybrids under rain-fed conditions in Benin. And finally, I would share about lessons learned from advancing this research study on MSVD and contribution of the thesis. So by way of a background, it looks a little bit small, but this is just showing the distribution of crops of the world. And in that corner, maybe I use my pointer. Okay, so this is maize. It's a very important staple in Africa. In fact, the third most important staple in the world. For people use it for fiber, for feed, and for food. It's widely grown in Africa, most consumed cereal in West Africa, but this particular crop faces major production constraints, both abiotic and biotic constraints. This crop is interesting because it thrives due to its ease of propagation. Everyone grows maize in their communities, no matter, not everyone, I mean, farmers, local people must just throw 1 or 2 seeds at the back of their houses, everywhere, because it's a food for livelihood, for, food security. It's a very important crop, widely adapted everywhere to most conditions. And it's a target by breeding programs for biofortification, so as to eradicate some vitamin A deficiencies and even improve it for zinc and iron. Yeah. But then we want to zero in on one problem, which is the maize streak virus disease. And why is that for maize? Because it is the most important biotic constraint of maize. This particular disease is economically significant to maize production in Africa. It's the only endemic disease to viral disease of maize. It's endemic to only sub-Saharan Africa, and it's caused by maize streak virus, which is a virus belonging to the family Geminiviridae, and it is a member of the maize virus genus. It is the type member because it's the most important member in that particular genus. It's vectored by a leafhopper known as Cicadulina, and it has a lot of species, Imbila, Noade, but Imbila is the most prominent vector of this particular virus. And the epidemics of this disease has been shown to occur every 3 to 10 years. It may not show up every time, but when it comes, it has significant effects on the crop production in any country where it is. The disease is actually aggravated by droughts or irregular early rains. So these are the predisposing conditions of the disease. First of all, drought is a factor, and as well as irregular rains. Whenever we have irregular rains, maybe like we have some climate change right now, where we're not experiencing enough rainfall, the disease can be present when you have susceptible varieties, as well as all the 3 important factors for a disease to occur: the host plant being susceptible, the virus also being present, as well as favorable conditions, which are now these irregular rainy season or droughts. This can cause the disease to manifest in any place. The symptoms of this disease include yellow streaking parallel to the leaf vein of a maize plant, severe stunting and bareness. You can even have intervenial necrosis, sclerosis, and eventual death of the plant. As I also said before, the age of the plant is a factor. If it's infected at 2 to 8 leaf stage, that's when it's most severe. After this stage, you may still have your 100% yield from your crop. So it's not always that bad, only when you have it occurring at this stage. The extent of susceptibility depends on, of course, I said before, the plant, if it's resistant, susceptible, the viral strain present, as well as the conditions, the climatic condition, usually drought. A little bit about drought. Yes, we know that it's taking effect currently, and it's costing the world about $37 billion per year. That's a USDA data. But then I come over to what I wanted to solve in the thesis. First of all, the quantitative property of this resistance. The resistance to the virus is of a quantitative nature. It has always been overflogged in literature that there's an overdependence on one particular chromosome to confer resistance to in maize, just an overdependence of particular MSV1 QTL, which is spontaneous trait loci, on in the use by the maize community breeding program in conferring this resistance to this crop. And that is a major factor, because if there is a breakdown, on this particular QTL, maybe a breakdown because probably the virus mutates or anything, we may not be able to protect the livelihood of people because it's going to ravage everywhere. So the literature, we have Ladijobi et al. 2018, Garcia Oliveira 2020, they have been stressing that there is a need to move to other sources of resistance or incorporate other sources of resistance to be able to manage this, and apart from just this major QTL. There's also a gap in knowledge of the number of QTLs that are already present and can be used, or at least what do we know from literature? What have we gathered so far? Because this research has been almost 10 decades long. They keep on researching on this particular disease and resistance for more than 10 decades. And so the question is, what do we have now? And what can we do with this information? So I tried to produce, or as I go along, try to produce a meta-analysis of already mapped QTLs controlling the trait of interest. And also the current status of maize, of the resistance to maize. What are we using in the production areas? What is the status of resistance to this disease? Their cultivars, their OPVs that they're using to grow. How resistant, where are we on that? The current, because much work has been done, a lot of varieties have been released, a lot of hybrids have been generated. What is the current status of the ones being grown currently? And what is their, how do we quantify their readiness to face any eventual breakdown of this QTL in the event that there is a breakdown? This information will be required for disease management, to optimize disease management, help plant health and productivity, and then avert any imminent yield losses in the event of an epidemic. So if there's an epidemic in the future, we need to know what to do to manage the disease. Besides, we have new sources of resistance currently. We have those from Ladijobi et al., like I said. He or the group tried to map some other QTLs to other chromosomes apart from that MSV1 on chromosome 1. So they mapped those ones and it's available right now. We also have those from Garcia et al., Oliveira et al. She and her team mapped other QTLs on other chromosomes as well. So now, if you want to utilize those information, those genomic resources, how do we utilize it? How do we transfer, integrate these QTLs into the local adapted cultivars so that we can, and from there, we have to then quantify the combined ability of these resistance sources when you combine them in crosses with the local adapted cultivars, as well as the heterosis of the inbred lines. Therefore, from there, when we quantify the combining ability and heterosis, we move over to estimating the yields, the agronomic performance, G by E, and all of that to be able to know how to proceed in release of varieties for farmers in West and Central Africa. So successful implementation of that meta-analysis will be required to serve to identify the consensus genomic regions. What I mean is, so we have so many QTLs available now. We have so many mapped QTLs, I may say. And so we want to know, are we going to use everything, or we're going to concentrate on a few major effects QTLs from these new sources of resistance? Therefore, the implementation of that meta-analysis was important to be able to develop markers that will now be used going forward in breeding. Another one is the baseline information that I had to go embark on to generate this information on the currently used cultivars in Benin's production system. So what is the resistance status of all these the currently grown OPVs and cultivars, as well as estimating this combinability when I have transferred the traits into the local adapted cultivars. I needed to estimate the combinability and then understand their interaction with environments across production environment at different agroecological zones. But then the central question, if I come back, the central question of this work really is what is the current situation of MSVD in Republic of Benin, and what measures are taken to improve and to build resilience of resource-poor growers in the event of an epidemic. Specifically, I wanted to, of course, again, know if there is a consensus at the molecular level for the newly discovered QTLs, and what are the current status, specifically the questions raised, combinability, the interaction across environments. The objective, principal objective, is to assess the status of MSVD, document coping mechanisms by farmers, as well as develop high-yielding MSVD lines that can now be grown in the community for sustainable production in Benin. Again, specific objectives: carry out meta-analysis of map QTLs, identify these regions, conduct a countrywide farmers field survey of MSV disease incidence and severity, and any other associated viruses of maize, assess the yields and agronomic performance of top cross hybrids developed, and then analyze the G by E interaction and stability of the improved hybrids. I hypothesize that there are no co-localization of QTLs conferring MSVD resistance across the genome of maize. That's a hypothesis. So all of these QTLs, if there is a co-localization of the QTL, and I will explain what that means going forward. And I also hypothesize that there is low prevalence of MSVD. I mean, with all of the hybrids produced, with all of the OPVs generated from the maize breeding program of IITA, of CIMIT, and being claimed to be deployed into, with all the donor monies that have been going into research so far in the last 10 decades, what is the status of MSVD in Benin? And also hypothesize that the elite donors, so these new sources of resistance, they are good general combiners. That's what I hypothesize, that there will be good general combiners for hybrids, for heterosis to be used to generate better producing, the right word is superior yielding hybrids. So I hypothesize that they are good general combiners, and that there are no interactions between the maize, the genotypes, and the environments, which we would see within the study. So that is that for a background. And thereon to the research status, the MSVD research status in Africa. That's a paper published in Theoretical and Applied Genetics. So firstly, I would like to say from literature that there are the prevalence of MSVD across Africa currently. MSVD is still affecting, when I say MSVD, maize streak virus disease, is still infecting and affecting farmlands across Africa. This is a accumulation of papers, research work, where they have gone through country surveys, they have done country surveys, but these four countries, I mean, five, you have Ghana, Togo, Ethiopia, Uganda, and Kenya, has seen a resurgence again of the disease. So they had curbed it before, and now there is a resurgence of the disease in the farmlands. These are all surveys, different studies over the years. So the objective of the paper was to dissect the outputs of the meta-analysis carried out on all mapped QTLs that are conferring resistance to these MSVD, new sources of resistance, and then to propose modern breeding approaches that can be used to introgress these QTLs into the local adapted cultivars. And I will stress that especially in a stress tolerance breeding program, because MSVD is an adaptive trait. It's not really a trait that there is major research on. Why did I say that? It's an adaptive trait because it is in the event of an epidemic. That's when you would need that resistance. But normally people go for drought, people go for major abiotic stresses like flooding. But when you know that there is a harm lurking around, there is a disease that in the event that there is an epidemic, all your drought tolerance, all your flood tolerance are not going to stand that disease. So I try to know propose breeding approaches that we can use to continue to introgress these very important QTLs, even when we are going through our normal major mainstream stress tolerance programs. We can continue to incorporate the maize-streak virus into, do not leave it aside because it's an important enemy lurking around, if you get my analogy. So the meta-analysis was carried out. The methodology is simple one, using the biomercator software. A software was used in doing the analysis, biomercator, and it entails putting together trying to gather some information like the 95% confidence interval of each of these new QTLs that have been mapped. Then you have to also get the lower, the upper limits, and then project all of these QTLs on a genetic map. And we use the B73 genome. The B73 germplasm, its genome was used to project these QTLs onto that genome in the biomercator software. At the end of the day, a total of 25 newly mapped QTLs, apart from the major MSV1 QTL on chromosome 1, was identified and uploaded into that package. So what I mean by that is that, so you have MSV1 as a QTL, which is the major resistance source of MSVD. So that is what have been overused. All these while breeders have markers for it. They're going to screen their lines, but then we're talking about conferring durable resistance to the disease. That means taking advantage of all the other minor QTLs that have been recently discovered, especially recovery resistance to MSVD. And then we got 25 QTLs from works only done with SMP QTL mapping, genetic mapping. Of these 25 QTLs done using SMP genotyping, we have 18 QTLs that accounted for 3.1 to 21% phenotypic variance for the traits. 4 QTLs were found on chromosome 3. Then we have 4 QTLs obtained were found on chromosome 3, while another 2 were found on chromosome 7 and 9. That's a beautiful story, knowing that we have the MSV1 on chromosome 1, and we have now new sources, they can contribute up to 20% on chromosome 3, 7, and 9, and that can be pulled in addition with the chromosome 1. And another information was that when we did this method, I want to show you that this is the genome of the B73 maize, and you have this QTL hanging around here, this second one, and another 3 here, 1, 2, and 3. These 5 QTLs had no co-localization. That means they were not, as you can see, they are far away from each other. They are not interfering, and there's no epistasis as such going on, and they could be narrowed down. When you transfer this information, you can actually then do the markers and use it for analysis. This means that each of these QTLs can be individually dispatched in addition to MSV1. All I'm stressing with the thesis, this work, is that we can continue, like they have said in literature, not to depend on chromosome 1, MSV1 and chromosome 1. So these additional QTLs can be incorporated by breeding programs and used in routine screening of their lines. And if it's not there, then we need to find a way to introgress more resistant sources so that we can be prepared for any eventual epidemics, the disease. Some approaches include, of course, marker-assisted breeding, which is using these 4 QTLs with high R-squares, phenotypic variation. We can target these 4 QTLs that had no co-localization among themselves on the genomic, on the consensus map. We can target them for candidate gene identification and develop markers that can be added to the MSV1. Also, genomic selection can be done. With the beautiful new strategies, genomic selection, you don't have to continue to have just 1 or 2 markers. You can pull together minor markers, minor QTLs, moderate QTLs, major QTLs in the genomic selection program as you are putting together your droughts and other things. So you can have enough things to go with. When you are selecting in your training population, you can pull lines that are more resilient, that can be more tolerant in the case of any eventuality of the MSVD. We can also have intermating of resistant sources in the magic population. We can try to do more of mapping magic populations in our breeding program than just biparental crosses, that are very limiting and will not be able to pull together enough favorable alleles for all of these stresses that we require. This is a bit of my approach in crossing when we have a magic population of 1, 2, 3, 4, 5. These are 6 parents. You have 1, 2, 3, 4, 5, 6 parents here. And with all of these, you can actually be able to then synthesize this population here that with genotyping and targeted genotyping, you can be able to synthesize and bring together favorable alleles that can now be deployed for use in MSVD resistance. Again, another strategy, a flow chart, trying to show how we can achieve this pattern. I'll move over to the survey since I'm pressed for time. I'll move to the survey where I did a countrywide survey also across all the major belts of Benin, and the objectives were to evaluate the MSV disease incidence and severity across maize belts of Benin, characterize MSV diversity and those associated with other viruses associated with maize, assess current situation of maize farmers in Benin in terms of their experience with the disease, as well as document strategies that they employ in trying to cope with the disease and manage the stresses. A survey was conducted across 6 agroecological zones. In southern and central Benin, we had 3 zones. In northern Benin, we had 3 zones. Excuse me. The study happened between the cropping seasons from June to September 2021, whereby I had to sample 20 plants for viral symptoms per field. And then we went through 114 fields as represented on this map. Everywhere across from north to south, 114 maize fields were visited and leaf samples collected for further analysis of the viruses we saw. These are only symptomatic plants across the fields. That's the questionnaire that was prepared and used. And when we got into a field, this is what we did. We tried to divide the field into 2 diagonals. So on this diagonal of the field, we take 5 symptomatic plants, 5 symptomatic plants, and we take another 5, and that is 20 plants sampled in the field. And then in each of them, that is 20 plants, 20 plants observed, while we take one leaf samples from each of them. So that makes us 4 samples, 4 plants that were taken from a field. 20 plants observed, 4 plants taken from a field. Data collected in every field we got to. We had to take passport information of the data, the GPS, the name of the farmer, and the location of the farm. And then we had to also assess farmers' knowledge of the disease. So we had to do a little bit of questioning about their knowledge, if they have seen it, if they know the virus, if they know the virus, the disease, and if they actually do anything to control whenever they see this disease. Regarding the results from our survey, we found that maize streak virus had a mean severity of 1.6 in this zone. That's the total of 240 plants assessed, so and so forth. We had 2.1. So when I say severity, that is a score between 1 and 5. Maize, 1 being not diseased and resistant or not showing symptoms, 5 being susceptible and showing MSV symptoms. And we see that the prevalence of maize incidence here, we had the highest zone, the zone having the highest amount of MSV is the west zone of Atakora. This is around the northern Benin, having almost 91.3% incidence of maize, of MSVD. Imagine a field almost covered with the disease. I mean, all plants showing these symptoms on them, and then the zone with the lowest being region of fisheries and vegetable crops. This should be zone 8 and southern Benin is having the lowest incidence of MSVD. Yeah, so this is a picture on field of what the disease looks like. We compared, so when we take a leaf sample, we try to do a multiplex PCR analysis and see if we have any other viruses present apart from MSVD. Some markers used for MCMV, which is maize chlorotic mottle virus, sugarcane mosaic virus. We even had markers from polioviruses used. We just try to screen every leaf collected for other viruses apart from MSV. And we found 2 interesting viruses apart from MSV. So we have SCMV present in about 10 samples. That's about 8.8% of the total samples collected, while MSV itself was found in 81 samples out of the 114 samples collected across the country, and that is about 71% of the total number of samples collected. As you can see, this is just showing the gel image that was used in running the multiplex QTR. If this band is the MSV, this is the MCMV, this is the SCMV. Interesting, why I'm dropping this slide, why I'm putting this slide up is because if there is there was no incidence of MCMV in Benin Republic. This virus is important. This maize chlorotic mottle virus. It's important because it is one of the viruses that makes up the network of maize lethal necrosis virus, the disease you see in Eastern Africa about 10 years or less than 10 years ago that wiped out maize farms in Kenya, in Tanzania. It's one of the diseases present, the viruses present apart from MSV, MCMV, and the maize lethal necrosis virus itself. So with this, because we don't have MCMV across Benin, that's a good one, knowing that if it's present and it's in line with, and it's present with MSV there, it will result in maize lethal necrosis virus. And that is a very lethal disease of maize, that combination, MSV, MCMV. But across all the places sampled in Benin, there was no presence of MCMV. That is important for disease management, especially with the authorities. In Nigeria, we have the quarantine services, and in Benin as well, there's a DPV. So it's important for these governmental officials to be very much alert about screening germplasms brought into the country so that we avoid boundary transmission of viruses. I'm very keen about this because my supervisor, Dr. Lava, is the global the IITA head for plant health, and we continue to screen every germplasm collected and are going out of the country for all of these viruses, knowing that in the event of any transmission, we begin to have issues, just the way we had the fall armyworm. If we were on alert, we would have maybe prevented the fall armyworm from coming into Western Africa. But today, it's prevalent everywhere. In East Africa, because of the MCMV, in conjunction with MSV, we had the maize lethal necrosis disease that wiped out farms. So there is need for health professionals to be alert using modern molecular markers, modern biotechnological markers to monitor plants and everything. And moving on, we try to check the diversity of the MSV samples. So we got MSV samples from different locations, and we tried to do a diversity study whether the MSV is still the strain known to cause MSV is still the strain present in Africa, in Benin or not. So we interestingly, thank God, I don't say thank God, interestingly, it is MSV A1 that is still present. And when you look at it in line with other viruses from other countries, you'll see that amongst the one we collected from our study, from MSV Bembereke, MSV from Basila, Materi, Tangeta, we see that when you look at this graph, amongst themselves, there's pairwise identity, high pairwise identity amongst the viruses. So, and it's known that it's A1, strain A1 that is still here. Actually, we have about 11 strains of MSV, and then we have 5 sub-strains of MSV-A. So it is the MSV-A1 that is the causal agent for MSV disease. So we are still in line. This is the result of the phylogenetic analysis. Also from this study, we had almost 11 virus strains compared with other viruses of other countries in the NCBI gene bank. And it's showing that it is actually clustering with all the MSV-As of other countries. That's one thing. Knowing that if it clustered with any other MSV, maybe G or B, that could be a problem, knowing that there is recombination event that could happen that would change, make the virus to evolve into something else that we don't have control of. So for now, in Benin, we are still working with MSV-A1, is still present and prevalent. However, we don't want that disease in the farms, but that's what is still present. Then we move over to a particular virus we found again in Benin, SCMV. SCMV was present in 2 communes, especially SCMV in Basila and SCMV in Bembereke. And using those 2 strains, we compared it also to other strains from other parts of the world. And we see a pairwise identity of less than a maximum 90, which is the blue region. That shows that it's same, that's a high mark. 90 is a bit much. So it's still the same SCMV we see in other regions of the world, although it's present in Benin and has not mutated or evolved to something else. Also, when we did the phylogenetic analysis, we see it clustering with MSVs in Tanzania, SCMVs that are gotten from Kenya, China, and interestingly, Ecuador, Mexico, Thailand is clustering with such SCMV strains that are found there. Then we have a new virus found in Benin, which is the MAYMV, maize yellow mosaic virus. That is the first report. This is the first report of MAYMV in Benin, and it's a polarivirus, although it is known to infect sugarcane, maize, and other things in Nigeria. But I mean, this is the first time the survey, I've not seen any other survey on maize in the viruses in the last 3 years for Benin. But with this study that was done in 2021, this was the first report in Benin to find this particular MAYMV. And we found it in 4 communes: Materi, Tengeta, Basila, and Bembereke. Okay, and we see it clustering to a large extent. This is between 98 and 99% identity with other MAYMVs of other countries. So, but we would not say it's not known to cause any effects, but there is no known effects, like there's no quantifiable effects of MAYMV in maize production systems. But however, we just have to make sure that we are on the alert in the event that this particular virus has any other effects in the future. When we surveyed the farmers, all from all communes that we attended, we're going through 19 communes across the country, which is 6 agroecological zones, which is 114 farms, maize farms. We try to check the incidence of maize per commune, of MSV per commune, per department surveyed. And we see it's most prevalent in all of these communes, Kandi, Gogunu, Materi, and Tangeta. These are communes present in northern Benin, at least. And then also in Bembereke, central Benin, as well as Basila, we are seeing MSV being more, the incidence that they're being more present than in the south. Every other commune with the bars below 20% are from the southern Benin. That means that MSVD is present more in the northern parts than in the south. And I would explain that by because of the drier regions in the north, and that can also be about the drought. We're talking about the drought, the drier regions, meaning drought, when there's less water, irregular rainfall as well. Maybe that's why the MSVD is more prevalent in the northern part than in the southern region. When we surveyed the farmers and their knowledge of the disease, we found that for where they get their seed source, where they get their seeds, most of farmers in Benin get seeds from, they buy their seeds, about 60%, more than 60% of farmers buy seeds, while about 40% save seeds from the previous seasons. In terms of preferred maize varieties, only one farmer, I would say it again, only one farmer grows hybrid maize in the places we surveyed across the country, while all about 113 farmers out of 114 grow local cultivars, OPVs. So that's an important statement, knowing that that means if farmers are using OPVs, we need to improve the OPVs for this disease. And even though we have to introduce hybrids, we make sure that we introduce what is adaptable, what can work for them, because they only work with OPVs. And so again, we talk about the MSV awareness, the awareness of the disease. We have about 60% of farmers saying that they are not aware of this MSV disease, while another 40 or 50%, 40-something percent say that they are aware, they have seen it. When we show them the pictures and samples, they say they have seen it. And then about controlling the disease, in case they want to make control, they say, no, all the farmers, in fact, all farmers here do not know anything about controlling the virus disease. So therefore, that means that there is much work to be done by extension agents, breeding programs, in order to push the knowledge and the control to the fields. And then finally, in chapter 4, I moved over to what I can contribute with this information I have coming from what genomics resources we have, as well as the farmers' interests, preferences, and what could I do? So the work has been published in this journal, but then the objective of this study, which is on assessment of the yields, analyzing the G/E and stability of improved maize top cross hybrids under rain-fed conditions in Benin. So the first objective was to evaluate agronomic performance and heterosis of developed top crosses, hybrids, and identify high-yielding genotypes for commercialization by SMEs, small and medium enterprises in the region, estimate combinability and heterosis of these lines used for the development of top crosses, investigate the relationship between grain yield and other traits, assess the effect of G/E on grain yield, and select top crosses with broad and specific adaptation across Benin. Lastly, I assess the representativeness and discriminative ability of test sites in different agroecologies. However, before I started the work, I first of all sampled all known adapted maize cultivars in Benin, all grown local cultivars in Benin. I sampled them and I tried to assess their status because I can't begin to improve on what I am not aware of, if they have the resistance or not. So this work was done at the Biology and Molecular Diagnostic Lab of the IITA units. I did it over 2 years. First of all, I collected 15 local cultivars, I got it from EMERAGHI in Benin. Yeah, then I also collected 2 MSV resistant inbred lines from IITA, which have these recovery resistance that I wanted to introgress there. Then also 2 source populations with drought tolerance and MSV. Then 2 MSV inbred lines from CIMIT, Mexico, were brought in. 1 MSV susceptible inbred line from Mexico also was collected. And then the phenotyping happened over 2 years, like I said, where we first of all read the vector that is used to transmit the disease. We screened for the MSV resistance in pots with 2 replications, testing them, with another pulcistine, which is a spreader plant. We use it to increase the quantity of MSV in the vicinity. So 1-week-old maize seedlings were artificially inoculated with these diseases using the Pseudomonas species as vectors. We scored for disease severity, percent disease recovery, area under disease progress score, and susceptibility scales and values. We even went ahead to test for the virus by using absorbance using ELISA method to check the absorbance at 405 nanometer and absorbance fold values, absorbance fold difference. That is the difference in MSV accumulation between the top leaves and the bottom leaves. This picture shows us, this is the first year, 2020, and the second year. Of all these 15 local cultivars grown in Benin, in the first year, they had, by the 6th week, even though the disease starts, first of all, being the disease severity is very high at the first week, but by the 6th week, they begin to decrease and even begin to recover from the disease sometimes. So, but at the end of the day, when we see the difference between week 1 and week 6, in the first year, they were really low. There was not much, that means they actually looked like they were resistant. But by the second year, this was the week 1 in blue, almost scoring 5. They were all very highly infected and showing symptoms. By the 4th week, by the 6th week, they were also, some of them with very high, they remained high, inoculated with the MSV. They still had that MSV load at very high proportions at week 6. But the difference as well between week 6 and week 1 is still showing that, yes, even though it's present, they are not significant as such. They're still going to be able to have moderate resistance to the MSV. So with that, I moved over to, since this just shows me that means there is moderate resistance in MSV in Benin. And that could be due to the MSV1 that was used to improve in the earlier stages of the maize breeding programs. So, but we are talking about MSV in other chromosomes, enhancing the MSV resistance using the newly discovered QTLs. So with that, I crossed the newly discovered sources of resistance with Benin's adapted maize cultivars. So in using the mating design line by tester and do across this season, making crosses from March to June, I took only 2 maize resistant sources, this and this from IITA. This and this from IITA, and then 11 white local cultivars or OPVs as lines, and developed 22 F1 hybrids. With that, including the pulcistine, which is a cultivar check, a susceptible cultivar check, I put them up for evaluation in the fields. So they were evaluated for combinability estimation and grain yield, as well as for G by E stability. This was done in 4 environments, location by year, using RCBD with 3 replications and with these specifications of the agronomic practices carried out. Data was collected on flowering traits, vegetative traits, yield and yield-related traits. And then analysis was done for combined analysis of variance, combinability effects estimation, mid-parents heterosis, correlations among traits, and then using the additive main effect and multiplicative interaction analysis and GG biplot analysis was used to analyze for the G by E interaction. Results. We see that environment was highly significant in the I'm looking for the word. It's highly significant in the expression of the grain yield traits, and in fact, almost all the traits. Environment, that means where we tested was sufficient to differentiate between genotypes. And again, the lines that were used, the combinability coming from the lines was significant, as well as the tester for grain yield. And then the specific combinability was also significant, and for environment also significant, for grain yield at least, which was the most important trait to farmers, although 100 seed weight is correlated with grain yield and antithesis seeking intervals. For the EME analysis, so there are 2 experiments, like I said, experiment 1 and 2. Experiment 1 tried to evaluate combinability using only the 22 hybrids developed. Experiment 2 added the pole 16 cultivar and used it to evaluate for what varieties are stable, what varieties performing better at what environments, that means the G by E interaction. And we see that both environments, genotype and genotype by interaction are highly significant in expressions of grain yield. The mean grain yield of all 23 genotypes have been shown on this table. What is most interesting is on this page, genotype 17 had the highest grain yield in all the environments across, even in the mean across all environments. It had the highest grade of 5.1 tons per hectare, as opposed to the Czech. This is genotype 23. 23 had a fairly high grain yield, although it's susceptible to MSVD. So we then moved over to estimating the GCA and SCA. I want you to note that these two testers that are bringing the resistance sources are of contrary GCA effects. So one is bringing negative GCA and one is bringing positive GCA. So when you use this particular tester 2, it helps to increase, especially the grain yield. It has a positive grain yield. It's in positive effects on the grain yield. While tester 1 would continue to drop the grain yields in your hybrid production. For the lines, only line 6. So these lines, their names are kindly, line 6 is DMR ESRW. It's an adapted cultivar, the DMR ESRW. That is the only line bringing positive, significant grain yield effects, GCA effect, combining. When you combine with T2, which is also positive, with these 2, that will be the one bringing you the most heterosis in your hybrid production. Specific combinability effects, there was no significant positive, but there were positive interactions, but no significantly positive ones for grain yield. And then lastly, the mid-parent heterosis and beta-parent heterosis, we only found on here. This is the most important one. L2, like I said, line 6 by tester 2 had a mid-parent ability of 29.1%. What does that mean? That means that if you combine line 6 and tester 2, you're going to have an increase of grain yield with 29.1% increase of your yield for that, heterosis being 29.1% increased. We try to estimate the different combiners who is bringing more alleles to the population. And we found that the OPVs do bring more favorable alleles to any especially the grain yield. It's bringing a lot of favorable alleles to the do I have what side? Okay. It's bringing a lot of favorable alleles to this interaction, to the hybrid production. Finally, for the correlation, we also saw that the grain yield did not have significant correlation with most traits. However, amongst traits like days to 50% silking, ear heights, and 100 seed weights, there was highly significant correlation between such traits, and they can be used for indirect selection for this particular population. The EME analysis, additive main effects and multiplicative interaction, showed that we have contributions of G by E up to the extent of 82%. That means that the G by E is doing more in affecting our work than we thought. And so we have to break down this G by E to know which environments can cause us to have better hybrid developed or a performance in grain yield than any other what environments are going to be good for screening, for bringing in, for production, and for expressing this high heterosis that we want to achieve in our breeding program. So we see that which one where what. That means this is a plot that shows us which genotypes are doing better at what environments. Which genotypes are doing better at what environments. For the tips of the genotypes at the tip of this pentagon or hexagon, we see that genotype 29, genotype 6, genotype 7, 13, 12, and 8 perform better at specific environments. For Seku, we have genotype 8 doing better there. For Masi, genotype 6 is performs best at Masi. And for genotype 29, for Dan, genotype 29 performs best at Dan. Ranking genotypes using the GGE biplots, we see that the ideal genotype that can do better or is stable across all environments is genotype 13, 15, and 11. These genotypes are doing best at Paraku and can be deployed for production in those agroecology around Paraku. Mean versus stability as well. This tool is used to assess what genotypes can give us the stable yields across all environments. What genotype can we use? Because this one performs at this stage, this one performs at this percentage at this stage, but which one would do best across all environments? So, and from this work, genotype 13, 15, and 11 are proposed to do best at all environments from this study. Relationship among environments shows that each of them are on different, and none of them there was no mega environments in this work, meaning that each of them were sufficient in discriminating amongst the genotypes. That's why you see that for Seku, we have genotype 8 doing best. For Dan, genotype 21 or 17 doing best. For Masi, genotype 20 doing best there at this environment. So our general findings are that environment is a significant effect, has a significant effect on grain yield and MSVD incorporation. There's a significant interaction between the environment and the lines for GCA or testers and SCA. that the parental genotype contributed more to heterosis, like the OPVs L6, the line L6 contributes more to development of hybrids than other lines, other OPVs. 4 OPVs actually had significant positive GCA effects for grain yields, possibly harboring favorable alleles for high yields and resilience. There was consistent positive MPH, which is mid-parent heterosis and beta-parent heterosis for grain yield, observed in L6 only, L6 by T2. So L6 is DMR ESRW by T2, this line from IITA. This is the best hybrid from this work and can be proposed for production across the environments of Benin. Some lessons learned as I round up is that screening of local cultivars and OPVs is important, and it can reveal, it was important in review that there is moderate MSVD resistance in the local cultivars. There is moderate, yes, I'm in my last 2 slides. Yes, I'm in my last 2 slides. Screening for local cultivars that was done at Benin revealed that there was possession of moderate resistance in the cultivars, but there is need to continue to improve using all these other sources of resistance, the newer sources. That we also found out that we also learned that making crosses, strategic crosses between newly sourced resistant parents is beneficial for introgressing recovery resistance to MSV, so in heterogeneous backgrounds. So when you call it strategic crosses, means that you have to be able to identify if you have good combiners or not before you make your crosses, and so that you can have excellently yielding hybrids from those crosses. That we also learned that exploiting heterosis from careful use of good combiners can bring about rapid yield advantages in our easy to formulate top rot hybrids. And this can give us a good potential for commercialization by the indigenous seed companies. Lastly, we learned that when we estimate the effect of G by E, we're able to identify stable performing hybrids that return predictable yields, ultimately important for food security for Benin Republic. And that when we leverage omics for trait discovery, we will be able to identify efficient alleles that can be used in manipulating for future strict resistance breeding. And lastly, disease management must be at the top of the table of health professionals, and especially the DPV and the quarantine services in the country, so that we can monitor newer, evolving, or emerging or re-emerging viruses in the system. Thank you for your kind attention.
Okay. Dr. Carlos, can you introduce Mary for us?
Thank you very much. I'm pleased to introduce to you Mary EMERAGHI. She is a Nigerian, and she was born on September 15, 1988. Of course, she schooled in Nigeria, where she obtained her first degree in combined biological sciences, majoring in biochemistry from the University of Nigeria, Nsukka, from 2006 to 2010. She completed her master's degree in crop science, majoring in plant breeding and genetics from the University of Benin, Benin City, from 2013 to 2016. She is an avid science communicator with aims to build capacity for young researchers and students, which led her to work as assistant lecturer in the Faculty of Agriculture, Benson Idahosa University, Benin City in Nigeria from 2016 to 2019. She was awarded with a scholarship under the EU-funded Intra-Africa Gene Mobility to pursue her PhD study at the University of Abomey-Calavi, with her research focusing on assessing the current impacts of maize streak virus disease on maize production system in Benin, with a view to propose a way forward for resistant hybrid development. Luckily, in July 2019, she was awarded with a grant from the International Foundation for Science to work on assessing the current situation of streak maize virus disease in framework of her PhD. During her PhD study, she supervised 2 undergraduate students and 2 postgraduates. She is currently a research associate at the Africa Rice Center Nigeria Station, hosted by the International Institute of Tropical Agriculture in Ibadan, where she provides breeding support in the rain-fed lowland breeding unit toward the development and evaluation of diverse type of rice breeding population for 28 African countries. Mary EMERAGHI is married and blessed with a set of twin girls. Thank you.
Okay, merci. We can sit down. Welcome to Prosaic Podium.
Chairs,
Yeah.
thank you so much. Can you hear me, please? Let me put on my video. Hello. Bonjour.
Bonjour.
Professor. Bonjour. Thank you so much. Oh, I hope you can hear and see me, please. Hello.
Yes, we can hear you. We can hear you, sir.
Oh, very well. Thank you so much, Chair. Mon Président, merci beaucoup.
Hey, merci. Okay, we will give who's the first? Okay, Dr. Caspar.
Yes,
Okay. Can you ask some question to Mary?
Thank you very much. Good morning, everyone.
Okay.
I hope you can hear me loud and clear.
Okay, after this one.
So I've got a few questions for Mary. Firstly, I want to congratulate her for putting together the nice thesis that I read. I enjoyed reading it, and I was also impressed by her presentation today. So I will start with the first question. Mary, you said that MSV disease is mostly endemic under water limited conditions. I would want to understand more about this scenario. What are the reasons behind the proliferation or the incidences of MSV disease under water limited conditions?
Can you answer?
I should,
Uh,
I thought you said questions, question one, okay. So the MSV is endemic to sub-Saharan Africa. Thank you. MSV is endemic to sub-Saharan Africa, and that is known in the literature, and then underwater limited conditions. So that is when the disease is expressed. The actual, maybe the science behind why the MSV virus thrives in dry conditions was not in the scope of my study, but this was what I gathered from literature, that the MSV would always manifest when there are three conditions present: the environment being drier, when you have susceptible cultivars, and also when you have the right strain present. So these three disease conditions would always cause the MSV to manifest. So this was what I got from the literature as to the very reason why it is always preferring dry, water limited conditions. I would have to delve more into that as a further study. Thank you, sir.
Thank you, Mary. I will move on to the second question. I understand that maize virus is transmitted by an insect, Cycadulina mbile, to be specific. My question is, what are the possible roles that may be played by natural enemies? By natural enemies, I'm speaking of pathogenic microorganisms or other insects. What are their possible roles in combating spread of the MSV disease?
Thank you very much, sir. So that is a possibility to use natural enemies of Cycadulina mbila to control the disease. But this particular vector, from my study, from literature, it's known to be migratory. So it's like a locust. It moves, it's a seasonal vector. It moves from maybe based on wind speed or season, it tries to migrate from one location to another. If you release natural enemies of Cycadulina, which has not been studied, I would say, from the best of my knowledge, I have tried to search literature if there is a study on any entomological study that shows natural enemies being used to combat Cycadulina. I have not seen. The only control methods have been to use the chemical method, which is sprays and insecticides to plants by host plant resistance, which is incorporating beta resistance for the disease. But as to natural enemies, which is other insects preying on the Cycadulina, maybe that would be for a further study, but I have not seen the literature to back that up. I don't know if I have answered your question, sir.
Yes, I'm happy with your answer. I would move on to the third question, which is more or less related to the question that I've previously asked you on natural enemies. I understand that some insects are deterred by some specific plant species. In the surveys that you did across Benin, was there any evidence of some specific plants that are being used by farmers in cropping systems to minimize incidences of the MSV disease?
Thank you so much for your answer. And sir, in my study, there were about 60% of farmers that actually recognized the disease, and 40% had not noticed, or maybe they just see the disease as abiotic effects or natural discoloring. They just say that, I mean, they have not seen this disease, and if they have, they just, because it doesn't affect so much the yield, so they don't go do anything about it. And I also surveyed them for if they ever control it by using any method at all, whether by planting or another plant to reduce. They said I noted that in a graph, if I can share my screen, that 109 out of 109 farmers have no control methods for MSVD yet, as at 2021. So I would not say that there is some crop or some method that was used by the farmers, because none of them control it, because they are not aware that it's actually affecting their crops. So just know that they have seen it occurring, but as to what to do about it, they just hope for the best and harvest whatever they can harvest. Thank you, sir. I hope I answered the question.
Yes, answered. And thank you for your response.
Thank you, sir.
I will move on to the fourth question. You mentioned that the majority of farmers in Benin use OPVs. And my question is, what are the possible reasons behind this current scenario? And my second question on this regard, as a breeder, are you comfortable with this current status?
Okay, thank you, sir. As to why they use OPVs instead of hybrids, I would like to attribute it to the costs of hybrid seeds in the low-income country of Benin. When I visited the what they call it now, they have their institutions that are in charge of seed sales or seed distribution to farmers. They say that the dynamics for the government to bring in and subsidize maize hybrids is still being worked out. They cannot say that there is no subsidy, but as at now, there is no modality to reduce the costs of hybrid seeds for farmers. So they would rather invest in cotton than in maize for subsidizing of hybrid seeds for growing by farmers. So they just resort to either saving their seeds or buying cheap OPVs that are produced in the country, whether it's resistant to MSVD or not. They just want to eat, and as whatever they harvest is what they can use. The second question, sir, could you remind me, please, sir? I'm trying to hold.
Before the second question, if the president allow me before the question, Professor Casper, if you can allow me before the second question, let me add to her response that there was a culture of seed distribution, dissemination, free seeds distribution to farmers in Benin, and it was mainly based on OPVs, not strictly local varieties, but OPVs, improved OPVs distributed for free to farmers. And that created a dynamic, and currently introducing hybrids into the system, it's a bit complicated because farmers were not used to buying seeds. But there is a new politics in place whereby new organizations are contributing to test hybrid seeds across the country. But yet, we still need effort to make that new dynamic work properly. So that's a contribution to the answer of Mary. Thank you, Prof.
Very much, Prof. Thank you for that clarification. I think she can answer the second bit of the question. If she is comfortable as a breeder with the current status of
Yes, sir. Yeah, so as a budding breeder, I would like to, first of all, I would be fortunate to contribute to the dialogue with the government to make how would I say now, subsidizing of hybrid seeds important because of the benefits of hybrid seeds. For example, the maize streak virus disease. If improving it would make the hybrid seeds more expensive, then we can make the farmers understand by enough advocacy, enough extension work, making them know why they should transition from OPVs or free OPVs to paid hybrid seed, knowing at least part of the payments, because the government can then match up with subsidy on the costs so that they can be prepared and have better yields, which means better income from their crop and from their labor. So I don't know if that, so that the discourse, the dialogue for me as a breeder would be more on the policymakers trying to do more with subsidization of hybrid seed than just leaving out the discourse just because of costs or you know, because of cost of the seed. It won't help the food security system of that country. They will continue to grow low yielding, cheap income crops than move up, come buy the seeds at a lower cost and have more harvest and feed more people and begin to export even for the country's foreign exchange and having more income. So that would be better for the farmers when they see that there is more, because they are business people. There is more when you go higher to hybrid seeds. I will be on the advocacy lane as a breeder, even with my science. Thank you.
Thank you, Mary. I'm
Chairman.
happy with you.
Okay.
I'll move on to my fifth question. I'm only left with one more. You mentioned that you crossed 11 OPVs from Benin and 2 MSV resistant lines from IIT. I would want to understand your intention. What is your end goal based on the decision that you made?
Okay, thank you. Having read the work of Badu Apraku et al., 2016, 2017, I think, he did the same thing by producing top cross hybrids from the same methodology and was able to synthesize high yielding top cross hybrids that could then be used for commercialization. So I thought to stand on the shoulder of such a giant, a maize breeder in IITA, and it's a cheaper method than other types of synthetic hybrid production. And because the seed companies will have to, first of all, maintain both parents, they have to keep on producing inbred lines or keep on making sure that they have pure lines of the inbred lines and then keep on not allowing the other parent, which is now another inbred line, to maybe break down or to become impure. So that is a costlier approach, using 2 inbred lines that you have to keep on maintaining and maybe even transition to other lines. But then using a cheap OPV adapted cultivars that are already fixed, and then only having to maintain 1 inbred line, or if possible, buy inbred, have an MOU with IITA to buy inbred line, and then just cross with local already fixed lines. So that it's a cheaper, cost-effective way of producing hybrids that you can sell the seed companies can sell higher. So with this method, top cross hybrid production, it's the most cost-effective pathway for hybrid development for seed companies than if they were to maintain 2 inbred lines. So this was my motivation, and standing on the work of Badu Apraku et al. 2016. I don't know if I answered the question, sir.
And thank you for your response. Related to this question, you selected 4 locations where you did your testing of the top crosses. Again, I would want to understand what informed your decision to choose those 4 locations. Were you satisfied with the outcome? Were they effective enough to give you the answers that you were looking for?
Thank you, sir. Why 4? It was mostly about my funding, which started in 2018. It wasn't as much to do as many locations as I would have loved. And then I had already started before I got the second funding in 2020 to augment my studies. So I had to do 4, firstly, because the locations were at the teaching and research farms of the units, the faculty's research farms, and they were across different parts of the country and spread across 2 agroecologies. So I thought they were enough to screen. Agroecologies, 2 different agroecologies, yes, secure enough in the research farms of GBios, where I'm doing my PhD in Faculty of Agriculture here. And then with funding issues, but on normal occasions, I should go for multiple locations across. I mean, we have 6 agroecologies of maize, major maize growing ecologies in Benin. Because of funding, I would have loved to do more, but I had to limit myself, even over the years, to be able to do more than 4 locations, maybe the next year, and others. All of this was based on funding. And this was a major factor to my not able to publish in some other journals than where I did, because they wanted more locations, more years, multiple years, to be able to properly ascertain if we have GBies that could explain the traits that I studied. So I would want to attribute it to funding than my own ability to test genotypes. I would love to do more. And maybe after this work, I could even test across the 6 agroecologies, but I have just only 4. This was a limitation. Thank you.
Thank you. Related to this, you chose to use randomized complete block design, and you were testing 23 genotypes. Do you think RCBD design was the best in relation to the other experimental designs that were at your disposal? Was it the best in terms of taking care of environmental variation?
The design accommodates a maximum of 25 genotypes, which would not, I mean, based on field heterogeneity, the gradient of fertility for each location, RCBD would go for a maximum of 25 genotypes in the literature, as opposed to alpha lattice, whereby you have more than 25, 30 genotypes, you can then go for 5 by 5, or I mean, some kind of alpha lattice design, 5 by 5, 10 by 5, 10, anything you want to do. But with 22, I still thought RCBD was within the RCBD's statistical rule to test with that design than go for something higher than that. It could still explain the diversity, or sorry, the differences between genotypes. It was just a matter of statistical rule or methodology Not that I would have tested for alpha. It was not up to 25 genotypes. So I thought RCBD would be able to cover for that heterogeneity. I mean, it's replicated per location and replicated per environment. So it's sufficient to explain the differences in the performance of genotypes. That was why.
Thank you very much. My last question related to the question that this aspect that I'm asking you. I noticed that on the trace that you recorded across the 4 trials that you conducted, you didn't score or you didn't measure for MSV resistance or susceptibility. And was there any special reason for that decision?
Yeah, yes, sir. Firstly, I chose to do first of all, for the scope of the PhD, and beyond I could do more, but for now, I chose to test under rain-fed conditions, optimum conditions was the first objective. This is optimum. The 4 environments are under optimum conditions, rain-fed. And if I were to continue, it would then be under stress, like MSV, under heavy hotspots of MSV across the nation. But for the scope of this work, I only stopped at rain-fed, optimum conditions. So scoring there, I didn't see more than 10% incidence of MSV. It was not significant to be able to do any work under rain-fed. I know that the disease manifests in droughts. So, but this is under rain-fed. So this could be a part of a work for later where we can then do rain-fed under these optimum and stress environments for MSV research. I mean, evaluations for further studies. Thank you.
Thank you very much. I'm done with my questions.
Thank you, sir.
Thank you.
Professor Caspar, are you
Hello. Yes, I am satisfied with the answers.
Okay. Thank you. Professor Igbopodion.
Thank you. Thank you, Chairman and the panel of jury. Thank you so much. Earlier on, I couldn't communicate. I didn't get the message, but with the excellent contribution and interrogation from Professor Caspar, I now understand. Thank you so much. First, I want to join the members of the jury to congratulate Mary for a very nice job done. I can tell you, while I reviewed the thesis, it was a really well thought out, well coordinated job done. From my end, this is excellent PhD research. And I understand that we have a scope, and within the scope of the study, I saw from the data, very nice job done. And for me, I was fully satisfied. However, there are always limitations. And I'm happy that answering Professor Caspar's question, she had acknowledged those limitations. And of course, that provides basis for further studies, further research. Thanks, Professor Caspar, for bringing that out of her. I mean, she was able to respond to that, and I'm very happy. Thanks, Professor Caspar, for that intervention. And Mary, thanks for your contribution.
Thank you, sir.
Now, as a breeder, I was really at home with you, going through your thesis. Mary, and I'm very proud of what you have done under the supervision of excellent and respected colleagues, Professor Achigandako and other colleagues at your renowned center. Let me drop this to the panel of jury. We are increasingly ever proud of your center and the influence of your center, the impact of your center on training really gladdens our heart, the African scientists, and we are proud of what you're doing. Please keep that up. Now, my question that's by the way of comment. Now, Mary, I want to ask you one or two questions and also drop a note for the faculty. It's very important as we go forward. I'm talking to my colleagues now at the faculty. You are addressing very important question that impact food security in the country. You are doing very well in addressing this. One point I want you to please consider for future study, if you are not already doing that, is to look at this same study under stressed conditions. Reason, number one, from the survey, you saw the prevalence of this maize-streak virus disease in the northern part of the country. And that tells, of course, you know, I'm sure the lower rainfall amount, raining days, the amount of rainfall received, that normally will predispose our maize varieties, whether OPV or hybrid, to, you know, the incidence of maize-streak virus. So that is really important. You need to go beyond the optimum condition to the stress condition, just as we experience in the northern part, number one. Number two, Burkina Faso, you need to watch the movement of materials, germplasm, across border. This is serious because coming from our experience, that is one issue, major issue. When you move materials across border, you could move them with, you know, the virus also. And then the control of the vectors. I thought that would be useful for further studies. That's my comment. I've looked at your methodology, very nice methodology, and I was very happy the way you defended your methodology when Professor Casper wanted you to speak up. Why you use RCBD and your model for us as breeders. This is the standard model for using EMERAGHI and G by E biplot to estimate. And those results are really great. Now, one question that I have for you, Mary. Now you've done your PhD, and now we want to translate our research results into impact, where our farmers can benefit and the country can also benefit. I mean, the issue of food security, we have more maize available for us to use. And even export, we need maize in Nigeria here, we need maize from Republic of Benin, so we can do trade. Now, what will be your recommendation for one, a big player in the seed industry that wants to be involved in this, a big player ready to invest millions of billions of CFA to establish a seed company? What will be your advice to such a big player in terms of varieties that will be distributed across the country? That's number one. What varieties would you recommend and why would you recommend varieties? Number two, for a small medium SME, a medium scale seed producer who, due to fund limitation, wants to play regionally, what variety will you recommend to someone who, for example, is in maybe southwestern part of the country? Somebody, say another medium player that wants to play in the north central or the mid country, the mid section of the country, and the one that want to play in the north. So you have small producers, small seed producers, one based in the far north, one based in the middle belt, and one based in the south. What varieties among these hybrids would you recommend? And why? Practical use of your research result. Thank you. Face me, don't look at your book. I want to see your face directly. Thank you.
Thank you so much. I'll look at the book for the questions. The first question was a big player in the seed industry, advice using my work. Firstly, as we noticed, the work has limitations in terms of the number of locations that it was tested in to prove, that's PROOF, to prove the validity of the hybrids that did best. So I would, first of all, like the big player to invest in R&D by doing more multi-locational studies of the 22 hybrids, actually, to actually run it in a deeper, in a more intensive manner. And then if L62 still is the superior, L62 is DMR ESRW by TZIL 032. If that is still the most superior across environments after the G by E interaction has been studied, then I would advise the big player to go ahead and promote by doing, going through the process of seed certification. You can then buy the, because there has to be some MOU between the breeder and the seed company so that the breeder could have royalties. Then he can take that technology, promote it, and then produce certified seeds that he can sell at large. And then it will be able to, it's a stable variety across multiple environments, so it could give this quantity of tons per hectare, and that could be what he could use to scale and produce a mass. So I would recommend that variety for the big player after intensive testing across many more environments. When you talk about sub-Saharan Western Africa, you can do that across countries, across production environment, both stress and optimum. And then having concluded with that arrow, and the first step will be the R&D, then that variety would then be chosen and used for going to the certification phase and then for production of certified seeds for distribution to farmers. For the small SME, the variety I would recommend also would be one that can thrive under low inputs of water, as well as that can do best under MSV pressure, under the MSV pressure, because northern Benin is more prone to the disease than the south. So again, there would still be a need for a little R&D in this, according to the capacity of the SME seed industry. And afterwards, out of the 22 hybrids tested under stress conditions, under low water, as in water deficient conditions, and under MSV stress, and other stresses actually, because MSV is an adaptive trait. It needs to be tested along with drought stress, along with biotic and abiotic constraints. And after having done a little bit under just narrowing it to northern Benin, he can then choose out of 22 hybrids which one, if L6T2 is still functional, then that would be my, because it is performing at 5.1 tons per hectare from my study. That is under optimum, which is using highest fertilizers, you're using optimum inputs. But on farmers' field, they don't have enough fertilizers, they don't have enough credits for business. So we need to make sure that it's tested under farmers' condition as well as on-station conditions before a choice can be made from this for under stress in northern Benin. In the south, where we have more rainfall, we may not need to do a more R&D, just any of the higher performing top crosses could be deployed since they have been improved for MSV and other things. They can be deployed. If it's performing well above 3 tons, currently the Benin maize system has a production limit or maximum of 1.06 tons per hectare under farmers' condition. So whereby a variety is scoring about 3 tons and more, that SME can promote such a variety for rain-fed without having to go through testing under stress and all that, because the MSV is quite, incidence is lower in the south and central than in the north. Thank you so much, sir.
Thank you. I think you may sit down, please.
Thank you.
You may sit down. Mr. President, and member of the jury, I feel very satisfied with not just the work, but the deep understanding of the principle and practice of what we do, indicating a very thorough training, and you have milled out of your university, your faculty, a very well-trained plant breeder and I see a great future, not just for her, but also as she joins us to mentor others. So I feel very satisfied. From my end here, congratulations. Thank you, Mr. President.
Okay, thank you, Wasai Pupodio. Ok, Professeur Adandono, nous vous invitons à nous faire part de vos appréciations si vous avez des questions à vous poser aussi.
Thank you very much, Mr. Chairman, of these examining boards. My name is Apollinaire. I'm from Apollinaire. I'm from the National University of Agriculture, which is located in K2, not far from Nigeria. Just a few comments before coming back to what I did about the valuation process. In terms of you're willing to have genotype which can be used across all ecological zones. I'm not sure that this would be. I'm using it, but okay. Yeah. So I was saying that I would like to make a few comments about what other examiners or reporters says about and you answer in terms of using genotypes across different zones, looking for resistance or looking for ability of a genotype to cope well with local conditions. With regard to photoperiodism, do you know photoperiodism? Yes, with regard to this and looking at the south and northern part of a country, I don't know. I don't think that because in the south there will be a lot of rain, and in the northern part of the country there will be drought or drought conditions, and I'm not sure that the same genotype can cope very well in terms of yield that we can get from the genotype across south to northern part of the country. But it could be also already good, but not easy to have genotype for the southern part of the country with a condition, local conditions of rain, and then a genotype for the southern part coping with the environment conditions in this area. Thank you very much again, Mr. Chairman. I have a privilege to evaluate these theses as a pre-evaluator. But first of all, let me congratulate the new vice chancellors of the University of EMERAGHI. And I'm grateful to the director of the doctorate school of this faculty for allowing me to be member of this examining board. Professor Achika, thank you very much for always having me as member of the board of examiners of all theses or most of the theses you are supervising. Thank you very much. That's very important for collaboration between university, between friends, between brothers and everything in terms of scientific knowledge. Coming back to the thesis, I appreciated very much the document. It is well written and readable. But for the first time when I was reading, I raised a lot of issues, right?
Yes.
And the new version now, I'm very happy for that, for the fact that you took into consideration or most of the point I raised to improve the document. For instance, I asked if you have references for the different agroecological zones you went through for the study. Did you have a reference now for that in the new version of the
Akosu et al. 2016.
Oh, okay, right.
Akosu et al. Is the reference.
Did you put it in the document? Okay.
Akosu et al.
Okay. Did you have a description of all agroecological zones also in? Because in the first version I read, this was not available in the document. Do you have it available now in?
Yeah.
Description of all agroecological zones.
Okay, so I was taught that when you reference on a when you give a reference to that, you're saying that the paper has made a full description. It had a table showing
No.
everything.
No. If it for my side, if you are publishing an article,
okay,
then you can just reference papers, not necessary to put details they put in the article, but in your thesis, I think you need to put details. And this is similar for the disease rating. Did you put the details about the disease rating in the
yes, I put the disease. When they have table 4.4, I don't know if you can go to page.
Okay. No, if it is in, then okay.
No, one of the characteristics of the environment.
No, I'm not talking
the description of the environment.
Talking about the disease rating.
No, that is there as well. See,
in which page is that?
Let me go there.
Yeah, but if it is in, it's okay. Because what I'm saying is like what I said for the references for agricultural zone, that you are not just referencing without putting the details. If you put details already for the disease rating in the document, that's fine. Okay. We move forward now. So, Mr. Chairman, I'm saying, I already have a look of the document, and like former professor said, I'm really satisfied because she took into consideration all points I raised. So here now, I will be just asking a few questions to interact with the candidates, right? Okay. So the first question is it easy to make a clear difference between the three virus diseases you find in the field, like maize streak virus disease, sugar mosaic virus disease, and maize clothing motor disease? Is it easy to make a clear difference between these three diseases in the field?
Thank you so much, sir, for this question. I think that it's a very valid question. I would say that on field, while sampling, we went for symptomatic plants, symptomatic for MSV. And when we sampled 20, when we observed 20, like I showed there, 20 plants, 10 per diagonal. After sampling and all, took 4 plants per location and went back to the lab. It was at the lab that using other markers in a multiplex PCR reaction that found that other viruses were present.
Okay.
So they were not symptomatic on the field,
Okay.
but they were present in the plants.
Okay.
So it was showing MSV symptoms, but it bore SCMV and the MAYMV discovered. That was an interesting one for, especially Dr. Lava. He would say, and you have no symptom of SCMV, yet it's present in the plants. So,
okay, but when we take these 3 diseases, which one is more prevalent?
Oh, MSV was the most on table.
Like showing your presentation. It's okay. I follow when you were presenting.
Yes.
That's fine. So you also identified then the different diseases, I mean, pathogen through molecular
methods.
Yes. Do you think that there is another method that we can use to identify disease in the field?
When there is a what?
Another method
Ah.
for disease identification instead of using molecular.
Well, recently there have been rapid diagnostics tools coming out from LAMP. There's something they call LAMP. I know it's the virology department now. I am a reader. So, but when I came in contact with them, they are always into detection, rapid diagnostic tools, bringing out whether ELISA methods, apart from the molecular PCR. So there is ELISA, there is LAMP assays. There are now RNA using, I mean, that's going again molecular. So everything is mostly molecular now.
Yes, I know. Okay. Do you know that phytopathologists talk about virulent and avirulent
strains?
Pathogen strains.
Yes.
Do you know that?
Yes, of course.
So do you think that for the same species, if we use molecular mean to identify virulent and avirulent strain, will we see any difference between those 2 pathogens?
I would say that when you say virulent, that means it's actually propagating the disease. Virulent may be present, but no disease. So that is the first way to even know. When you see that, oh, on my plants, I mean, no yield loss. First of all, there is no yield loss, even though the virus is present.
Yes, that's what
there is no yield loss. Yeah. So if there is a way to differentiate, that's what we
use molecular way.
Molecular
to identify.
Yeah, markers are particular for each of the strains. So like MSV A1 has markers for MSV A1 and other markers for MSV A2, and even other markers for MSV B to K. So you see that in the lab there at IITA, they are specific for specific strains. Okay. So the markers are present, they have it as per routine work because they must screen for MSV, even though they screen for other. And this MSVA, remember, the markers are for maybe the resistance, which is MSV1 as well. So they are only limited to MSV1 resistance for MSVA1 strain. What of other types of resistance that are coming up now so that you can improve and make the resistance durable? So that's why I'm advocating for markers for other resistance, other strains maybe, but the strain important for MSV is MSV A1, and the markers are present already.
So for you, is there any need for molecular people when they finish identification of a strain? Is there any need for them to go for Koch postulate?
For what?
Koch postulate.
Koch postulate.
Do you know the Koch postulate?
The Koch postulate that says that the three environments must be present. Is that what? Am I right? The 3 favorable conditions will be present for a disease to manifest. Is that the postulate? Koch postulate. I'm not very familiar. I'm not a virologist.
Pathologist or you are
I'm a plant breeder.
You are a plant breeder, not plant pathologist.
Working on a disease resistance breeding methods. Yeah.
No, I think this is.
I just dabbled and I got to learn a lot more.
I think this is very important for you because you are a
To
genetician.
be a biologist.
But you are, yeah, you are not far. You are not far from plant pathology.
Forgot.
Yeah, I only.
Every biologist.
I am trying to remember.
Okay, so Coach postulate is I'm in the field. That's why I was talking about a virulent and virulent strain. I'm in the field and I find a plant which is diseased. And I try to isolate microorganisms that I found in the environment of the plant. You see? And then I find two, A and B.
Okay.
Maybe one is a virulent and the other one, the second one is virulent, I don't know, or both. Now I need to conduct this test, which also called pathogenicity. You know pathogenicity?
Yes, yes.
Okay, so I need to check if the strain I got of a microorganism I isolated from field, from disease plant, if this microorganism is causing the disease I observe in the plant, I observe the symptoms in the plant in the field. So I need to pure culture the microorganism, inoculate it to a plant that I'm sure that is free of disease, and I look at if any symptoms appear from the inoculated plant.
Yeah.
If there is any
a reappearance of
I again isolate it
yes.
and have a look of now the specific this is my question. Do you think that we can do this apply to virus? And is it possible to apply to virus?
Yeah.
No, it's not possible.
It's a living organism, isn't it?
If it is possible to apply
although it's innate in nature, but when it comes in contact with a host, it becomes alive.
Exactly.
Yes.
When it's come to the contact to the host, that is the difference where we have an obligate pathogen. The virus is an obligate pathogen. So only we test it when it comes to the contact to the
host. Yes.
So we always say that for virus, we cannot be difficult to follow the postulate rule, but a modified rule is possible to test for pathogenicity of the virus. So I was sure, but I understood that you molecular biology people, you don't like to struggle for pathogenicity. Okay. Okay, so you did not carry out this. Okay, another question is, you know, there are 3 types of vectors carrying viruses causing disease. How many do you know them? 5 vectors which carry viruses to cause diseases. They are non-persistent, semi-persistent, and persistent.
Yes.
Right?
Cicadulina is a persistent one.
This one is what?
A persistent one.
A persistent. What is the persistent?
Vector? So according to the literature, it must have the virus in its guts at all times
and keep it permanent.
And keep it permanently. Yeah. Continue.
So
it's a persistent.
A persistent virus.
Yes.
Okay. Okay. So now, if you ask you, I'm so happy that you did this work in Benin. And my question is, Benin and Nigeria, they are just close. Why you prefer to come to Benin? You were saying that there is a resurgence of the disease in Ghana, Benin, and Kenya, and not in Benin. But the disease is there.
Yes, it's there.
So the first question is why did you prefer to come to Benin? The disease is in Benin, Nigeria also.
Yes. My being here is it's just a scholarship.
Scholarship.
Yeah, it was not by choice to say I want to go to Nigeria. It's the scholarship that I didn't even choose what country.
You are forced to do it.
I had to choose 3 out of 5 countries.
So initially, the closer to your country.
So when I chose 3, I just woke up to the email that Abby posted to Benin. I did not choose to be as such, as it were. I was just assigned for one of the three choices I made.
That's good. So if we ask you the mean incidence of a disease in Benin, how can you answer
The mean
incidence of the disease?
It would vary
it
will vary by zone, sir.
By zone.
By zone, because as we have 6 zones, I had mean incidence on the table there. In northern Benin, it's 91.3. 91.3% for MSV. But in the southern, the zone 8, it's 10.3 or 10.2%. Atakora. So it's varying like that from 10% to 91%, and it's by zone. So I wouldn't say there's a mean across the whole country. There's just per zone. Yes.
I'm interested in knowing if there is any link or any correlation between the incidence of a disease in the field and the disease, the yield losses. Did you make any correlation between, to say that if the disease incidence is 90, for instance, then the losses in terms of yields?
That data would have been important in the survey if I had took it. I didn't actually take the costs, the yield. Because when I ask a farmer, what is this? How many tons do you produce per year, per season? They keep saying maybe they say 10 bags of this. They describe the bag. They can't measure by kg or by hectare. They just say, oh, I can get 10 of this in 1 hectare. I can get 3 of this in another hectare. But to say that because of MSV, they can get 2 of this, so no, they don't know. They just only harvest what they can harvest and take it home. So they wouldn't attribute it to MSV. I have to have a full study to then, in real time on farmers' field, know the impact or effects of MSV on the costs, whether it is yield loss, whether it is higher prices, whether anyone, on the costs for farmers now. So I would go to a hotspot, allow the farmer to crop for the season and know and quantify the yields. As opposed to an optimum environment where the farmer also grows and then gets the yield. I would need to follow that through for every location, then be able to quantify the loss by MSV. The loss by MSV. That's when I can make a proper correlation. But to say a figure to you, sir, I don't have data to say that.
So by yourself, you did not
by myself. But
you know, generally, screening to check if
a work by Martin and Shepard, 2009, was able to cost MSV loss for South Africa, SSA, Sub-Saharan Africa. And they estimate that because of MSV, we have almost between $120 and $480 million lost for South Africa, SSA, because of MSVD. They were the only first work that has costed MSVD loss, the cost of MSVD on African maize production system. So that's what they say, and no other person has costed it per country, per region,
Okay, because I'm afraid about 1% from Atakura, where the incidence goes to 91%. I'm wondering if
it will be interesting.
People in this area, they are still eating maize.
It will be interesting to quantify.
But what I know from my experience is viruses, depending on the strain. Otherwise, viruses in maize. We can see maybe symptoms in the field that how this comes to yield is not clear. So it may be a good study if you can relate.
And also to note that MSVD is just a cause. Epidemics, the epidemics of it occurs every 3 to 10 years. And it's only when you have an epidemic that you have significant loss.
Yes.
It doesn't always result in loss. Exactly. In fact, at low as 17% loss, if it even happens. Yeah, and then move to 100% loss. Yeah. So that's why we call it an adaptive trait. Yes. Important and adaptive trait. Yeah.
How long have you?
Oh, yes. The disease recovers. It's actually plants recover from the disease after some time, especially if it is not susceptible, if it's not infected at the early stage. Like I said, the disease occurs more or would lead to loss when it was infected at 2 to 8 leaf stage of the plant. If it's infected at a higher age, the maize will give you 100%. It wouldn't affect the yield.
You mean in maize, eh?
Maize will produce 100% yield if MSV infects after that young stage. It won't yield the result to leaf. It's only at the early stage of the maize that there will be a loss from 17 to 100% loss. Yeah, there won't be loss after that stage. If it infects at maybe flowering stage, it won't give any loss. Yeah.
Good. How long have you been in Benin?
I came in October 2018.
Not just come today, tomorrow you leave to Nigeria. How long have you been?
I was in Benin
You
for
stay
a
in
full 2.5 years.
Huh?
A full 2.5 years before I
Okay.
moved to IIT.
Full.
Full.
Okay.
2.5 years or so.
So you can speak French?
Not really. Just a bit.
So, comment ça va? You
Ça
don't
va
know?
bien. Et vous? Ça va bien.
Monsieur le président du jury, encore une fois, merci beaucoup pour les autorités de l'Université d'Abodo-Calavi pour m'avoir toujours à côté des éminents professeurs comme vous pour apprécier donc, les thèses du professeur Achigan. Merci encore une fois pour cette collaboration que vous développez souvent. J'ai parcouru mes préoccupations. Encore une fois, je suis assez satisfait donc, de ce que l'étudiant a produit comme document. Au départ, quand j'avais lu, j'étais resté un peu sur la fin. Mais toutes les corrections ont été prises en compte et le document est bon. Merci beaucoup, monsieur le président du jury.
Merci. Merci, professeur. Merci pour tout ce que vous faites à nos étudiants. Alors, nous allons prier le professeur Boucan Dantin qui fait office d'examinateur dans ce jury, de nous faire part de ses appréciations d'une manière générale. S'il y a des questions à infuser. Allez-y. Vous avez la parole.
Thank you. Thank you, Professor Florent Kunom, President of this jury. Well, I'll start it. Will you please sit down, Mary? Yeah, I will start expressing my sincere gratitude to Professor Avelesi Félicien, rector of this university, for having allowed me to be a part of this jury, member of this jury. Because I have already been giving back to my parents after serving the university and the country for almost 40 years, 38 years exactly. But due to some maybe circumstances, or I don't know the reason why, I still continue coming, collaborating with GBAUS, this lab and other labs at the Faculty of Agronomic Science. My sincere thanks to the doctoral school. Okay, they are the one in collaboration with Professor Achigandako have decided to to call me to be a member of this jury. And I'm very grateful to Florent, my friend. Florent, I call him Florent. Here he is the president, but he remains Florent for me, my sincere friend. I really appreciate the a comment and by the three reporters who previously worked on the quality of this work, the work by Mary, and the co-directors on this work. I'm really grateful to them, because what we are having today is just the sum up of the work they have done, each of them at a different level. Having said that, I will start commenting on the quality of the work, which is a very good work. The reason why I'm part of this work and I'm interested in this work, one of the reasons is that I am an entomologist, and we will have not this maize streak virus disease without the expression of the vectors. So the Cicadolina, which are vectors of this disease. And because I'm an entomologist, that's the reason why I'm really interested in this work. And for the story, the very brief story of this work, I remember in the year 2020-21, when the little girl, she was at that time a little girl. Now she has grown up. She has grown up. She is not a little girl anymore. And I even heard that she is now in the same social group. We are, Mary and I are in the same social group. I have a beautiful pair of twins, okay? A boy and a girl. And she joined the club by being a mother of twins, okay? Beautiful girls. So that's the reason why we have something in common that you will never have. You, all of you, not you. Okay, good, Mary. So, examining the quality of your work, there are some, minor corrections to work on, when you take back and the copy I have with me. There are some minor corrections. And, the very first correction that really strike on me is the cover page. At the cover page, when you look at a reporter number 3, that's not his name. It's not his name, okay? His name is totally miswritten there. It's not Adandono, but it's Adandono, okay? That's a very, well, I call it minor, but it's a major mistake. So, other mistakes are, when you take the work is, something is missing in your work, is the autobiography. Okay, you should put the autobiography at the end, okay, of your work. If Dr. Carlos have not presented your autobiography, I will not really be able to know, when from, the beginning to the point where we are now, what has been your achievement. In terms of quality, I found also the figures. When you go to figure 3, for instance, the quality of your figures is not good. And when the quality is not good, okay, if I were, I were Mary, I will not say credit photo to Mary. I will just leave it like that. Because now you are emphasizing that you are the one who took those bad pictures. And for the quality of your work, it's not good. It's not good. But another mistake that came, not minor mistake, but major mistake, is the name of some scientific, the writing of some scientific names of crops and insects. When you write, okay, Zea maize, you know, I found it at many places in the document. Zea maize, we have Zea, which is what? Zea is what? The genus, yes. And maize is the species. The species should always start with small letters. It should not appear in your document that you have some typo mistakes like that. Because when I sometimes consider what you are now, if you wanted to apply for a job, you can apply for a job as entomopathologist. Entomology, pathology combined. You call yourself a breeder, okay? You call yourself breeder now, but let me ask at this point, please write it down, a question on this. I assume there is a big difference. Now I'm in the content. I assume there is a big difference in expression of the disease, not only with agroecological zones, but mainly with the type of variety. And yesterday, because of this defense, I went to the National Plant Protection Service and I talked to the head. And I asked to the head, how do you find, what do you think of the maize streak virus disease presently in Benin? So what he responded to me is not what you said. He said, we have 3 categories. And at these 3 different points, the expression of the disease is completely different. We have the hybrids, okay? We have the composites variety, and we have the local varieties. And for him, you know, the expression of the disease is important on local varieties, okay? And the hybrids, when you do not have a hybrid where the resistance of the maize streak virus is not, okay, you do have the very great expression of the disease, but you did not really emphasize, you know, your study putting a light on the local varieties. But do you agree with him? That's a very first question. Do you agree with him that the expression of the disease mainly depends on the local, the quality of the variety we have? Okay, that's my very first question. When I go to further onto the work, I heard you put an emphasis on the maize yellow mosaic virus just occurring in 4 different communes. And you know, the interest I have in this is that, is there any possibility, is there any risk that we have a distribution or an outbreak of that MAYMV disease in the country? because that will be from 4 communes to the 77 communes we have. You know, the step is not very, very big. The gap is not very big. What do you recommend? Because before coming here as entomologist, as a lecturer at the University of Abomey-Calavi, I was a principal staff at the National Plant Protection Service. That's why I always go there. I still maintain a good collaboration with them. So what do you think, you know, about that possibility, and what do you recommend, you know, for the small farmer, the smallholder farmers to not contribute into the spread of the disease? Yeah, another question is, well, I already comment on that. But from your work, it appears that you have acquired a very good knowledge of the country Benin. And I assume that you have a better knowledge now of Benin than Nigeria. Okay, you will tell me if I'm right, if this is correct. And building on the question of Professor Adandono, I have a position now, to work in Benin. Because you have a very good knowledge of Benin, I have a position at JBIOS. Are you ready to move from Nigeria to Benin and to take that position? Let me know what you think about it. And we'll start the process and the procedure immediately. And next week, you will move to Benin. I've been really impressed, Mary, by your oral presentation, even though it was still a bit long. It was a big long, 1 hour, okay? We asked you to remain in 45 minutes. And in other circumstances, you can be allowed to remain to give your presentation in 15 minutes. So you have to be able, as if ever you were declared now at the end of the morning, okay, doctor, okay, in what? And that's still my other question. Are you doctor in, well, plant breeding? Are you a breeder? Are you an anthropathologist? Are you a virologist? Okay, you will let me know exactly what you will be. Because I have a colleague in the jury, okay? He is like a chameleon. He is not an entomologist. He is not a pathologist. You know, sometimes he says he is an entomopathologist, but he will tell us exactly what you are. You cannot be entomologist and pathologist, Professor Adandono. Okay. Now, Mary, when you're reading your work, you know, my questions are a little bit at random, and you'll try to see which order you put in answering the questions. I realized that, you know, in your work, you directly jump onto the virus, okay? You did not put just one sentence or one paragraph to specify, you know, the impact of other diseases onto to maize. It's straightforward. You start, well, talking about, it's very good, okay? But I could have asked, would it be possible for you or to some other candidate to conduct the same work on maize and on another important disease on maize, okay? Because it's not the virus is not the only one, you know, problem we have here in terms of maize production. Maize is, I agree, effectively the staple food, a very good staple food in Benin, specifically cereals in Benin. This is the main one. Okay, when we go to root and tubers, and we can speak about cassava, but maize is a very good staple food. And that's even the reason why we do have the center of excellence, okay, on maize in Africa. When we consider different crops, you know, Benin is, we have implemented, you know, by CORAF and others, the center of excellence of maize production in Benin. So I really appreciate, once again, your work has told a story, okay? And you beautifully put this, okay, by saying that it's a beautiful, you said it in your presentation, it's a beautiful story that you have presented. And when I consider, you know, the meta-analysis of maize virus disease, that's the where you say that your results revealed a beautiful story. And I really appreciate this. Really appreciate this. And I congratulate you for that piece of work, which is a very good piece of work for you and for the student, the candidates who will come after you to work on this same subject of something very close. So that's it, and well, to complete, to finish. I recently read a yield assessment of new strike resistant top cross maize hybrids in Benin. And that work, that piece of work was by EMERAGHI and al. 2014. It was a very good piece of work. And I consider this that a kind of, okay, one of the very great papers you have produced. And I'm very proud of you. So throughout the papers, you know, the documents, very, very small mistakes, very small corrections that I will invite you to make to really complete and finalize the presentation of your work, because it will stay, okay, in our library, okay? Other students will take your work, you know, as a reference. So you really need to work on that so that we have a very good piece of work. Good? So I think I already asked my questions, Mr. President, and they were put at random. It's not an interaction with the candidate like other examiners, other have done, but I wish, you know, she gave me, okay, responses and to these different questions so that we consider this as complete. Thank you, Mr. President.
Thank you.
That's all for now.
Thank you so much, Mr. Examiner. The first question, do I agree on the expression of the disease, MSV, on local varieties? I would agree. I agree to that because, yes, you're also correct about hybrids also being susceptible to the disease if it does not have the resistance put in it. So wherever, whether hybrid composites or local cultivars, as long as the resistance is not in there, the disease will be expressed if it meets a susceptible, the strain. If it meets the strain on the field, if environmental conditions are favorable, which is drought, drier areas or less rain or unpredictable rain, whenever any cultivar, however the type, hybrid, composite or local cultivars, as long as they don't have that resistance, When they meet these conditions, the disease will manifest. When the vector brings the disease, there is no MSV resistance. It's going to manifest in the cultivar. So I agree that it's more drastic in local cultivars than in other improved or higher cultivars. So I agree. Then, is there any risk of an outbreak of MAYMV? And what do I recommend for smallholder farmers? Yes, the authors, Yahaya et al., 2019, who first discovered the disease in Nigeria, in sugarcane and in maize fields, has also attributed to the fact that, yes, it's not the impact of that disease has not yet been, it's not drastic, it's not very yield devastating as maize street virus, but it's something to watch out for, since it's a virus of maize and it surely negates the yield of maize. So for Benin, there could be, like I'm always saying, the plant health professionals will need to continue to monitor the inflow and outflow of germplasms and do periodic detection studies or periodic diagnostics of the maize production zones to be sure that all these viruses are not spreading or are not causing much harm than we can control. You know, there is something, we know that there is an economic threshold for every disease. So if it is going beyond the threshold, then they have to come in with disease-resistant hybrids or varieties or OPVs that the farmers can turn to for such a time. But for now, there will be a risk, of course, if the virus sees favorable conditions to express itself. But as of today, there is no outbreak of MAYMV. There is no prevalence of such or incidence of any outbreak, but it's something to watch out for by the health professionals, the plant health professionals of the nation. Yeah. For farmers, for spreading the disease, since, as I said, it's not as high as that of MSV, which is also dependent on the vector, but MAYMV is not vectored. It's not a vectored disease. So they would need to make sure that they use clean seeds, they use good quality disease-free seeds to plant. That's what they can do on their part, because most farmers save their seeds for next season and want to continue to use their seeds for next season. What if their seeds are not kept in good storage conditions? They could propagate diseases for the farm. So I would advise smallholder farmers to continue to plant disease-free seeds as much as they can, so that they can curb any, whether MAYMV, any other disease, they can curb disease spread as much as possible. To work in Benin, yes, if I'm given a good position, of course, I'm open to, I'm still attached on a contract basis with Africa Rice up till December. So beyond December, I may speak to that more firmly. Thank you. Impact of other diseases to maize. Oh yes, I would then need to rework the PhD, the thesis, to be able to have a page, a paragraph or two discussing on other diseases of maize apart from MSV before I then move into MSV. Yes, and then I'll take up maybe the corrections you say about the paper, the yield assessment paper. I would like to then know the different things. Thank you.
Non, je crois que c'est bon. Je suis satisfait. Thank you, Mary. Thank you. Keep on really the good work you have been doing, and try to initiate to coach some other young students and candidates in following your steps. Even though you are here or over there in Nigeria, or maybe in a couple of months, well, in California or some other places in the world. Yeah, merci. Thank you so much. Okay. Good, Mr. President. Merci beaucoup.
don't know if there is a co-directors online or not. No. Okay. There is what was comment ça va? Au CLB, Kuma, are you online? No.
Good morning. I'm Dr. Kumar here.
Okay.
Yes, I joined a bit late. I'm following the question and answer interactions. Thank you for the opportunity and congratulations to Mary on successful completion and hopefully this defense will go well. I can tell you she is very passionate in training people and passing information to others, so she will be a good mentor. And I'm sure she's going to give more opportunities for other students to follow her path and succeed well.
Do you have some questions for her?
I don't have any specific questions because I mean I have seen her work and the theses and all, so it's a very good work. All I would like to say is that I just pick on the last interaction point on the maize yellow mosaic virus. It was detected in even the first report from one of our students from our lab in Nigeria. It is so far known to not to cause any symptoms in maize, so it is economically not significant, as Mary mentioned. We have no idea about whether it spreads through seed, because by default it belongs to polyrovirus. That group of viruses are not known to spread through seed. And then it has vectors. They have aphids, so likely to have some other weed hosts which may be harboring, and from there it is going to maize. So maize is not a primary. There must be some other hosts which support that virus. But as Mary mentioned, it is important for us to pay attention and not to neglect it. But whatever the information we have so far, it implies that it is not causing any damage to the plant. Yeah.
Okay. So we'll ask Professor Achiga to give us some.
Thank you. Thank you very much, Mr. President. I would like to recognize the authority of the University of Abomey-Calavi and the postgraduate school for allowing the defense of Mary. I would like to thank all the partner universities, the University of Calabar in Nigeria. Thank you, Professor Peter Dion, for being present. This is the second time we are sitting in the same examination board. I wish we could continue the collaboration on co-supervising our students, and I believe that this will come as we continue working together. I would like to recognize the authority of the University of Zimbabwe. Thank you, Professor Caspar Kamutando, for being present on this examination board, but also for evaluating, assessing the report. Thank you very much. And the National University of Agriculture in Benin, thank you, Professor Adandono, for your scrutiny on this dissertation, and recognize also the collaboration of Professor Eme Bokono-Ganta, my professor and also mentor. Thank you, Mr. President, for the respected work you have been doing so far. This work has been co-supervised with Professor Oselebe Happiness at the university, at the Ebonyi State University in Nigeria, but also with Dr. Lava Kuma, head of research in virology and molecular diagnostics at the International Institute of Tropical Agriculture. Thank you very much, Dr. Lava, for being present and your contribution to the quality of this work. In fact, Mary is a scholar of the Intra-Africa Mobility Project known as JANES. JANES was managed directly from Ebonyi State University. And contrary to what Mary said, she was not imposed to Benin. She was the top candidate coming from Nigeria. And all the top candidates from Nigeria were allocated to Benin. That's why she was here. So congratulations, Mary. You never knew that you were the top candidate from Nigeria. I can tell you today that, yes, you were the top candidate from Nigeria. And in October 2018, when Mary received the information, she did not even wait for a travel notice or even a flight ticket before dropping herself in this lab here without even stamping her passports. So she had to travel back and do the formality before starting really the work. But immediately recognize the competencies and ability of Mary. She has a very writing skills and also a good analytical skills. That's why very early in 2019, she was involved in co-organizing the first conference of the African Plant Breeders Association in Accra, Ghana. Mary contributed to assess the abstracts. She contributed to organize the logistic, and beautifully, we all traveled from Abomey-Calavi to Accra to attend that first conference. And that laid the foundation for the second conference in 2021 in Kigali. But because of the COVID-19 virus, it was not possible to organize a group travel to Kigali. Then Mary participated online, but meanwhile, she took part in participating in the student wing of the EPI-BA. Currently, we are organizing the fourth conference in Zimbabwe at Victoria 4, where Professor Caspar is the main local organizer, and I believe Mary will be in Victoria Falls, and will be lucky to meet in person Professor Caspar, who is one of our top breeders in Africa. It's important to recognize that before even finishing her scholarship here, Mary traveled to Ibadan, Nigeria, where she worked with Professor Lava and got the opportunity to interact with other people, where she got a position now, which is finishing in December, as she said. But my prayer is that she continue interacting, working in the CG system. And I would like to plead that Dr. Koma continue to monitoring, so that this strength, competence that we have in Africa be groomed, promoted at a very good scientific level. I believe that Mary has everything needed to be a top scientist in the continent, and whatever each of us here can do to promote her will be necessarily welcomed. You will continue being a member of GBIOS wherever you are. She was lucky to raise funds for the lab through the IFS project. And this is something that I will invite all the students to do, try to raise funds to your organizations, because through those funds, you can also be nicely promoted. So thank you, Mary, for everything you've done for this lab. and we need to continue working together in years, months, and days to come. Thank you, Mr. President. Don't have any question for her.
Okay. Now we can move to another side for the deliberation.
Okay. To Professor Caspar, Peter, and Kuma, we'll move you to the examination room, and we'll meet there. Thank you very much.
Professor Caspar, Professor Aikopodion, and Professor Kuma, are you online?
Yes, I'm online.
You want to?
Yeah. Yes, please.
Yes. You want to deliberate in French, the official language of Benin. Madame Mary EMERAGHI a abordé un sujet d'actualité et d'intérêt sur l'état actuel de la maladie de la striure du maïs au Bénin. La candidate a présenté avec aisance son sujet et a répondu aux questions du jury avec satisfaction. Le jury a délibéré et a accepté le travail de Madame Mary EMERAGHI et le jugeant digne de porter le grade de docteur de l'Université d'Abomey-Calavi en spécialité ressources phytogénétiques et protection des cultures avec la mention très honorable et félicitations du jury. Très honorable et félicitations du jury is the highest, highest distinction of our university. Mm-hmm.
On behalf of Professor Oselebe and Professor Lava Kumar, I'll
Yes.
give you this as a recognition of your excellence. Welcoming to the big family of daughters of the University of Abomey-Calavi.
Thank you so much. Thank you.
This is a blessing from the whole family of the university, and it will bring you joy, happiness, progress in your professional career. Thank you.
Thank you. Amen. Congratulations. Congratulations.
Congratulations. Great achievement.
Congratulations. To God be the glory. This is beautiful. Congratulations. Congrats.
And also congratulations from me. Well done.
The ceremony is over. Meet you next time.
Yes, thank you. So I will be dropping off. And Mary, enjoy today.
Enjoy, Mary. Bye-bye, and congratulations.